Good morning everyone. I am Poonam Lahoti and we are starting with biology topic today. The topic name is nucleic acid. Nucleic acid are of two types. One is DNA and another is RNA. DNA ribonucleic acid and RNA ribonucleic acid. Now you must be having the question which has come first, whether it is DNA or it is RNA. So I would like to tell you that RNA came first in the evolution. Means when evolution was taking place, RNA came first in the evolution. Now, but today we are concerned with actually DNA. Okay, so that is deoxyribonucleic acid. So let's see the discovery of DNA. The discovery of DNA started in 1869. See, 1869, Friedrich Michel has started the discovery of DNA. But shall I tell you something? Friedrich Michel कोई लेना देना नहीं था with the discovery of DNA, because he was not discovering the DNA. He was actually concerned with pus cells. The pus cells, we are having pus in pimple. The pus cell are nothing but dead WBC cells. And when he was studying that cell, he realizes that this pus cell is rich of phosphorus. That pus cells were rich of phosphorus and he named that as nuclein. He named that as nuclein. As it was present in nucleus, whatever the cell he studied, he named it as nuclein. After that, after 20 years of that, we see 1869, he has discovered that phosphorus rich something is there in the nucleus, that is nuclein. After that 20 years, 1889, Richard Altman has discovered that the nuclein is acidic in nature. See, 20 years gap is there because the technology was not that much sufficient, and that is the reason after 20 years he has discovered that nuclein is acidic in nature, and so he named it as nucleic acid, and this is how the nucleic acid name came into existence. But after that, in 1953, see, in 1953, based on X-ray diffraction, two scientists, Franklin and Berkeley, see, हमने आज तक पढ़ा है कि डिस्कवरी ऑफ डीएनए जो है वो वाटसन एंड क्रिक ने की है, बट आई वुड लाइक टू टेल यू एक्चुअली जो रिसर्च हुआ था, वट एवर द रिसर्च हैपेंड, इट वाज डन बाय फ्रैंकलिन एंड विल्क्लिन एंड फ्रैंकलिन हैज डन सम रिसर्च by the process of X-ray diffraction, and they have discovered some paper. और problem is that बोलते हैं ना नसीब, so the same has happened with them कि वो जो papers थे वो गलती से लैब में छूट गए, और जब गलती से पेपर्स लैब में छूट गए तो वो किसी ने चुरा लिए. अब ये चुरा के वो बंदे ने जाके दे दिया टू वैटसन एंड क्रिक, and then Watson and Crick studied that paper, research on that, and they have elaborated the concept, whatever was discovered by Franklin, they have elaborated the concept, and on basis of that they have given us double helix structure of DNA. It was a two-dimensional, see, you should know that it is a two-dimensional structure which is given by the James Watson and Francisco Crick in 1953. But the Nobel Prize they have got for them, see, Watson and Crick को ही Nobel Prize मिला, then they got the Nobel Prize for the discovery of double helix structure of DNA. So let's see what this double helix structure is. Structure of DNA. DNA is a deoxyribonucleic acid, we know. Now DNA is a polymer of nucleotides. DNA is a polymer of nucleotides. So let's see what is nucleotide. See, from the name you can see deoxyribonucleic acid. See, deoxyribo means it's a derivative of sugar, ribose sugar. Ribose is a pentose sugar. So you can see here it's a pentose sugar containing 5 carbon. 1, 2, 3, 4, and CHO is that is 5th carbon over here, to which the phosphate group is attached. Okay. And And nucleic acid, because it is present in nucleus and it is acidic in nature, so the name deoxyribonucleic acid. Remember this. So sugar, it's a pentose sugar, and you can see on the first carbon there is an attachment of nitrogenous base. On the first carbon there is attachment of nitrogenous base. We will see what are nitrogen bases and all. And on fifth carbon you can see I have written P, that is phosphate group. So phosphate group is attached to the fifth carbon of the sugar, while nitrogen base is attached to the first carbon of the sugar. Okay. Now the sugar and nitrogen base. So can I say this? Yes. The sugar and nitrogenous base together we called nucleoside. What it called? Nucleoside. But if we add phosphorus in that and phosphate in that, you can see sugar plus nitrogen base plus phosphate, they form nucleotide. And polymer of nucleotide is nothing but DNA. What is DNA? It's a polymer of nucleotide. It means this whole structure is one nucleotide. This whole structure is one nucleotide, and many nucleotides come together to form DNA. It's a chain of nucleotide, that is polymer of nucleotide. Let's see which are the bonds present in one nucleotide. We are talking about one nucleotide. In one nucleotide, let's see which are the bonds present. You can see here, see, this is sugar, this is nitrogen base, and this is phosphate group. The bond which is present in the first carbon of sugar and nitrogenous base is called glycosidic bond. The bond which is present between sugar and nitrogenous base is called glycosidic bond. This is formed by the release of H2O. By the release of H2O, glycosidic bond is formed. The bond which is present between sugar, fifth carbon of sugar, and phosphate group is called phosphoester bond. Is called phosphoester bond. What is phosphoester bond? It is a bond present between the fifth carbon of sugar and phosphate group. So phosphate group is there, so phospho, and it's an ester bond. Okay, so it is phosphoester bond. Now, as I told you, this is one nucleotide. If I see, so can I say in one nucleotide there is a glycosidic bond and phosphoester bond, right? Now, coming to the second nucleotide, the same way we have taken one more nucleotide. And what is the bond between these two nucleotides? In between the two nucleotides, you can see fifth carbon phosphate is attached to the third carbon of the first nucleotide. Means this is second nucleotide, this is nucleotide number 2, this is nucleotide number 1, and fifth carbon phosphate of nucleotide 2 is attached to the third carbon of nucleotide 1, and the bond which is formed in this way is called phosphodiester bond. The bond which is formed is called phosphodiester bond. See, if somebody asks you which is a bond formed between the two nucleotides, you will say it is phosphodiester bond. And like this, many phosphodiester bond forms and the chain proceeds. Okay? Now, let's talk something about nitrogenous base. Nitrogenous base are of two types. Nitrogenous base are of two types, purines and pyrimidines. Okay? Detailing we are going to do in 12th standard, but right now you should know some basics, so I am telling you purines and pyrimidines. The detailing of this structure and everything we are going to do afterward in the next lecture. But you should know there are two types of purine, adenine and guanine, and pyrimidines are thymine and cytosine. See, in DNA, adenine, guanine, thymine and cytosine, these are the four nitrogenous bases are there. But if you see in RNA, adenine is there, guanine is there, cytosine is there, only the thymine is replaced by uracil. The thymine is replaced by uracil. This is the difference between DNA and RNA. In RNA, it is replaced by uracil. So thymine is not there in RNA because uracil is there. Okay. Now, the two strands The two strands of DNA are anti-parallel. What does it mean? I will first explain you what is 3 prime, what is 5 prime for this. Now you can see here when this attachment took place, here the third carbon is not free, it is attached with the fifth carbon. Which carbon is free is fifth carbon. And here on the last, when this chain happens, on the last which carbon are free, from this it is getting decided which is the third prime and which is 5 prime. For example, suppose the chain has end over here. So fifth carbon is free and bottom 3 carbon is free, third carbon is free. So we can say it is 5 prime to 3 prime. We can say this chain is from 5 prime to 3 prime. If it is in another way, then if third carbon of the upper one is free like this and the fifth carbon of the lower one, then we say it is 3 prime to 5 prime. Okay, so we can say it is 3 prime to 5 prime. And if you have observed here, this is 5 prime to 3 prime and here it is 3 prime to 5 prime. As these two strands are running in opposite direction, we call DNA is, the two strands of DNA are anti-parallel to each other. The two strands of DNA are anti-parallel to each other. Now, if we talk about these two strands, we have seen that the two nucleotides are attached to each other by phosphodiester bond. But what about these two strands? These two strands are attached by nitrogenous base to each other and the bond is hydrogen bond. They are attached to each other with the help of hydrogen bond. Okay, so two strands of DNA are attached to each other by hydrogen bond. And where the hydrogen bond forms? Between the two nitrogenous bases. Now, if the bond is there, adenine pairs with thymine and guanine pairs with cytosine. Adenine pairs with thymine and guanine pairs with cytosine. Adenine pairs with thymine with 2 hydrogen bond and guanine pairs with cytosine with 3 hydrogen bond. It means if guanine cytosine amount is more, that means the bond will be more, hydrogen bond will be more and the stronger will be the DNA with strands. Okay? If adenine and thymine will be there, then the only 2 hydrogen bonds are there and that is why that DNA will not be that much, that 2 strands are not be that much stronger. So, it decides how strong will be the bonding between the 2 strands. So, in guanine and cytosine there are 3 hydrogen bonds present, in adenine thymine there are 2 hydrogen bonds present. Okay? Thank you very much.